Hello there. My name is Paula. I am an assistant professor of pathology in the School of Medicine. I'm originally from Argentina. I have a master's in genetics from the National University of Misiones, and then I moved to the United States to pursue my graduate work. I got my PhD working on cancer biology at the Weill Cornell Medical College in New York. And I worked in a metastasis lab at Memorial Sloan Kettering, where I develop models, systems to study metastasis to different organs and characterize mechanistic programs that led to organotropism, so colonization of the brains and the lungs. My postdoc was in the area of immunology. So my lab right now is interested in how the immune microenvironment regulates tumor progression and metastasis. We focus on different clinically important problems. One of them, and the one that we focus most of our time, is the understanding of what are the mechanisms that facilitate metastatic progression, in particularly to the brain and the lungs. But we're also interested in understanding more about the crosstalk between different cellular compartments in the primary tumor and how the primary tumor can exert systemic effects that will then predispose cancer patients to metastatic dissemination. There is also another area of clinical importance, which is the transition between benign lesions in the breast and invasive cancer. We do have a very large population of people with benign lesions in the breast, but only some of them will progress. However, we can't really tell who is going to have cancer and who isn't, or how to treat this patient population. So these are the three areas that we try to better understand with the hope of identifying either mechanisms or molecular mediators that we can target. We collaborate with the pharmacy school to test molecules and medicines that can actually modulate the plasticity of the different cells in the tumor stroma to make them reprogram between something that was helping the tumor to now something that will fight the tumor. We mainly work with transgenic mouse modeling, but we do a number of other complementary studies. We do flow cytometry, we do immunohistochemical characterization of the tumor tissues, we do ex vivo analysis of the metastatic organs, we set up ex vivo functional assays between different cell compartments of the tumor. We're also doing omics, so RNA sequencing and single cell sequencing to understand what are the molecular or signaling events in the different components of the tumor microenvironment. And finally, we validate our mouse studies in human samples that we get from the clinic. The last thing that I wanted to mention is that all of the people who have trained in the lab, we've been open for about 5+ years, and everybody is continuing in the science field. Some of them have gone to companies, and some of them continue in the academic background. And we also have had lab members from different places in the world. We have a variety of sources of funding, and so I'm happy to talk to you this afternoon, or just reach out to me by email or via Twitter. Thank you so much.